Okay, so this is going to be joint work with Julia Schiffman, Dave Swafford, and Jing Peng, and I'll sort of tell you about the particular parts that they've worked on as we go through. So I know this in this group we all know what a phylogenetic tree is, so I won't quite start there, but I am going to define the two different types of phylogenies that I'm going to use throughout the talk in case people aren't used to thinking about the differences between a species level phylogeny and a gene level phylogeny or gene tree. So when I talk about a species tree, what I mean is a regular, sort of normal looking phylogenetic tree, except that the nodes represent speciation events. So in this example, there are 3 species, A, B, and C, and 2 speciation events. So, going forward in time, we have a speciation event here marked with the horizontal dotted line where A becomes distinct from the ancestor of B and C. And then here's another speciation event where B and C become distinct species. And the time t between those will be something important for us to think about when we start to think about probability models in this case. So that's the species tree. And then we can think of a gene tree as being the particular phylogenetic history for an individual gene sampled somewhere in the genome. And we think of the gene tree as being constrained to evolve within the species tree or within the speciation process. So what I've drawn now is a gene tree nested inside of the species tree. And the idea there is that I have sampled sort of 1 gene copy from species B, 1 gene copy from C, and 1 copy from A. And I'm interested in knowing when those gene copies shared a common ancestor. So if I just look at the samples from B and C, I can sort of trace back and see that here's the common ancestor to that lineage. And I'll note that that common ancestor has to sort of occur inside the species tree. So those 2 genes can't share a copy down here somewhere because we're assuming that after speciation, the two species are now distinct and there's no gene flow between them. So that's an assumption of the species tree. And that means that these gene ancestral events, these internal nodes in the gene tree, which I'll probably end up calling coalescent times, these coalescent times have to occur sort of further back in time than the node times of the species tree. So one possibility then is that B and C share their common ancestor in this interval of length t, and in that case, the gene tree and the species tree share the same shape because B and C are most closely related, followed by A. Another possibility is that the common ancestor of B and C does not occur in this interval of length t, and instead those 2 gene copies remain distinct during that time period. And then we get back here to the root population, and we see that there, the lineage sample from B and the lineage sample from C share a common ancestor, in which case we get a gene tree that matches the species tree in terms of topology, but is different from this gene tree in terms of where this common ancestor coalescent event took place. And then there are a couple of other ways to do things. We can again not have the common ancestor occur in this interval, but now when we get back into this ancestral population, it could be that the lineage sampled from C coalesces with that sampled from A. And now the gene tree is actually different than the species tree because in the gene tree, A and C are most closely related rather than B and C as in the species tree. And then there's one more possibility, which is that A and B coalesce first, followed by C. And again, we get a gene tree that differs from the species tree. So what you can see is that for this simple case where we only have 3 tips, there are 4 possible gene tree histories. So by history, I'm going to mean the case where we keep track of both the topology and the interval on the species tree where the coalescent event occurs. So these 2 trees at the top here are distinct in terms of their history. They share the same topology, but they're distinct in terms of their history because the first coalescent event occurs in the interval with length t for this one and occurs above the root for this one. And then there are 4 histories and 3 topologies. And if you start to think about this, you can sort of think what happens when we go from 3 species to 4 species. So here's a 4 species species tree. And so in all of the subpanels, the species tree is the same. And actually the gene tree topology is also the same in all of the panels, but the histories are different. So we can sort of look at what happens. And it happens to be the gene tree topology that matches the species tree. So the first coalescent event should be between C and D. So the first interval that can happen in is this one, okay? And then the coalescent event with B could happen here, and then the final coalescent event. So this first one is the case where all of the events happen in the first interval they possibly can. But we can think of other ways of arranging this. For example, this one could not happen until the next most ancestral interval, so we get this particular history. Or we could move this one up one, we get this history. We can move them both up one, or we could move everything to above the root. So for 4 species, we now have 5 histories just to match the species tree. So we went from 2 histories to match the species tree to 5 histories to match the species tree. And hopefully what you're thinking now is this seems bad, and this table sort of gives us an idea of how bad this is. So this is for differing numbers of species, and then for both the asymmetric species tree and a species tree that's as symmetric as possible, how many histories are there for the gene tree that matches? Okay, so this is just for the gene tree that matches. And you can see that, you know, when the numbers of species aren't very large here before this starts to look like a huge nightmare in some ways, because there are very many ways to fit a matching gene tree into the species tree. And then remember that in general, our goal is to estimate the species tree. So we're actually also interested in how many species trees there would be. So it's sort of, you know, we've taken the regular phylogenetic complexity problem where there's just lots of topologies to look at. We've made it worse by trying to fit a topology into the topology, right? And now we have lots and lots of ways to do that. So I just want you to keep in mind how rapidly this grows as we think about the rest of the talk. So now I want to talk some about data. Ultimately, I'm going to work on estimating speciation times today. So we're going to think about having data that's informative about the speciation times. So let's think about data. So we have now, you know, the genome, we have chromosomes, and the idea is that different locations on the chromosome will have different gene trees. So this is a nice picture that I stole from a publication in Nature, and it just sort of shows for human, chimp, and gorilla that at one particular location we might have the relationship where human and chimp are most closely related. That's what we believe follows the species level relationships. But other locations in the chromosome might have one of the other arrangements. So here's human and gorilla most closely related. Here's chimp and gorilla most closely related. So when I get a sample from the genome, I'm actually going to have data from a lot of different gene trees. But what I observe are not the gene trees, right? We don't go into the lab and derive gene trees. We go into the lab and we get sequence data. So our model is going to be that the sequence data comes from the gene trees, and the gene trees come from the species tree. But our problem is that we have sequence data and we want to estimate the species tree, and these gene trees are unobserved in the middle of that process. Okay, so I think I have a picture of that. Yeah. So here's the full data, the full model that we want to think about. We have a species tree. The species tree generates gene trees at different locations along the chromosome. And then the DNA sequence data arise from the gene trees. And our inference problem is to take the DNA sequences and go back to the species tree. Okay. So I'm going to think about doing this kind of inference in a model-based way. So I'm going to tell you just the names of the models without giving you very much detail about the models. So I'll be really happy to tell you all about these in detail if you want to know, but it'll take a while. So I'll just sort of give you the idea, and then we'll see how we're going to use it. So for this first part of the model, from species trees to gene trees, we're going to use the multi-species coalescent model. And the big thing to just keep in mind for now is just that means that the times to coalescent events follow an exponential distribution. So it's straightforward is what I want to say. The model is not exceptionally complicated. We just have times to events being exponentially distributed. The rate varies depending on how many lineages we have. So there's some bookkeeping, but it's not a very complicated model in that sense. And then for the second piece, the gene trees to the DNA sequences, we'll just use the standard continuous time Markov models that we usually use for the nucleotides A, C, G, and T. So things like the Jukes-Kanner model, the general time reversible model. So this is just all the standard models that we typically use. We're going to think of our data, our DNA sequence data, as being a set of IID alignment columns from the model that I've just described, this two-stage model. And our goal will be to estimate the speciation times for a fixed species tree. So we're not going to worry about searching for the best species tree. We're going to worry about trying to estimate speciation times on a tree that someone gives us. So let me define a little bit more about what I mean about the data. So we're going to assume that we have an alignment, and I just stuck with this example of 3 tips for now. Oops. And we're going to look at a column in the alignment, and I'm going to call that a site pattern probability. So this particular column is one in which human has nucleotide A, chimp has T, gorilla has A. And so I'm going to call that a site pattern probability, ATA. And I'm sorry, that's a site pattern, ATA. And I'm going to be interested in the probability of that site pattern arising under this two-stage model, the coalescent and then the nucleotide substitution models. And I'm going to assume that each of the columns in the alignment is an IID independent, identically distributed observation from this two-stage model. So I have a nice sort of standard statistical problem. So if you think about this, we might first say, okay, this doesn't seem so bad because so there are, if you only have a 3 taxon tree, there's 64 possible site patterns. You've just showed us these 4 possible histories. You can just write down all the probabilities. No big deal. Then you have a multinomial likelihood function, and it's the regular phylogenetic problem because it's this multinomial likelihood. And so you could find the MLE, or you could use a Bayesian approach. You could, get whatever you wanted. The problem is that would still be hard, right? Because if we go past 3 tips, we're still going to have to do a search through the space of species trees. And we know that that's hard even under the simpler case where we're just trying to estimate a gene tree. But the very bad news is actually that computing those site pattern probabilities is really tough. And in fact, we don't have nice closed form expressions for those for more than 4 tips. So I want to try to convince you why that's a hard problem, because otherwise the things I'm doing to get around that won't be so interesting if you think you can do this problem, because this would be a better way to do it if we could do it. So let me try to convince you why it's sort of hard to get good expressions for more than 4 taxa. So here's what we would need to do. We have this 2-step model, right? We have gene trees coming from species trees. So this piece in blue, F, is the probability density function for a gene tree history with its associated coalescent times given the particular species tree and set of speciation times. So that's the part that comes from the coalescent model. And then there's this is the usual sort of what people have called the Felsenstein likelihood, the probability of the data at the tips of the tree given the gene tree, both the topology H and the set of times associated with that. But what I don't observe is what the gene tree is. I observe only the sequence data. And so I've got to integrate over all possible gene trees. So I'm going to take a sum over the histories and an integral over the times associated with each history. Okay, so still maybe not so bad, but now let's go back and realize that, the number of terms in that sum, if I have 20 tips, is like this, right? And then each one of those we've got to worry about the limits of integration for each one of those, right? Because the time is going to occur within that interval of length t. And so somehow I've got to keep track of all those limits of integration. And so it's actually really hard to figure out how to write this down in general or how to program it in general. Okay, so right now we have expressions for 4 tips. And that's what I'm going to work with today is what can we do with the expressions that we do have for trees with 4 tips. Okay, maybe a good place to pause. Does anyone have a question before I keep charging along? Okay, I assume everyone's good. Again, feel free to speak up if you have questions. Okay, so Julia Schiffman and I spent quite a long time actually doing this. We just enumerated everything for 4-tip trees for both the symmetric and the asymmetric tree, just to kind of see what things look like. So we used what I'm going to show today are results with the Jukes-Kanner model, JC69. And I'm going to show you some things with the symmetric tree. We have all the same things with the asymmetric tree too. In this case, there are 9 distinct site pattern probabilities. And what we sort of mean by that is if you have sort of A, C, A for the tree that I've drawn here, that would be the same as C, A, A. Because I've assumed that this time is tau 2, and so they evolve independently, so it doesn't matter which one you give A and which one you give C to. And then there's other similar things that come from symmetry of the Jukes-Kanner model, like the pattern AAAA has the same probability as TTTT under Jukes-Kanner. So while there are 56 possible site pattern probabilities, you can reduce them down to a set for this tree of 9 distinct site pattern probabilities. So that makes our lives a lot simpler. And so what we can do is write expression, a nice expression actually, for the probability of these site patterns under this two-stage model. So this is the model where the gene trees come from the species tree with the coalescent model, and then the data come from the gene trees under the Jukes-Kanner 69 model. And so this IAIB, ICID is just, these are 4 nucleotides, so the probability of AAAA given this species tree with these particular set of speciation times. And the X's in here are just exponentiated versions of the times. Okay, so this is a nice expression. These C's are just constants that we can compute, but I like to show them to people because we spent a lot of time getting them. So here they are. So here's the set of, here's the set of C's that go with the XXXX pattern, the XXXY pattern, XYXX. Okay, so there they all are. They're things you can compute. The theta is just the parameter in the coalescent model, which I didn't define carefully, so you don't need to worry about what that is. And then mu is just the value for the Jukes-Kanner model. That'll be 4/3 for the Jukes-Kanner model. So you can evaluate these. They evaluate to numbers, but we have general expressions for them. Okay, and even better, you can write that in matrix form, right? So if we define our parameter vector beta to be, these x's, which are the transformed taus. So think of these x's as the speciation times. They're just speciation times transformed. Then we can write the equation that C, that set of things I just showed you on those 3 slides, times the parameter vector beta, is equal to the probabilities, the vector of the site pattern probabilities. Okay, so shown here, the vector of those 9 things. And that means then that I could solve for beta, right? And so it turns out that there's a unique solution. And so now I have expressions for the branch lengths in this case. And these expressions are just functions of the site pattern probabilities. Okay, and it's actually great because that is something, if I knew those P's, I could just compute this. It's not an optimization problem even, like you have in a regular likelihood setting where you have to find the values that optimize the likelihood. This is a formula. If I knew those P's, I can just plug them in and I'll get estimators of the branch length. So it's a really nice setting to have. And then you can so these are X's, these are the transformed branch lengths, so you can just transform back. So that suggests actually that we could use this for inference, because while I don't know these P's, I don't know the probability of getting constant site pattern because I don't actually know the taus. If someone gives me an alignment, I can estimate the probability of the constant site pattern, right? I'll just go through and count how many times did I get all A's? How many times did I get all C's, right? So very easy, right? Computationally efficient, very easy to scan an alignment and count up how many times you get each of the site patterns. So this can be done very rapidly. So that suggests that we could use this for inference. And so we'll just take the P hat vector. So this is just the observed counts. We'll plug it in for P, and we'll get our estimates. And so we did a simulation study to see if this was going to perform well. And so we used James Degnan's program COAL to generate the gene trees. We used seqgen to generate sequence data on the gene trees. And we experimented with, I guess, 2 different sample sizes, so 10,000 sites and 100,000 sites, and set our taus to be 1, 2, and 3 in coalescent units. And the first row is the data-rich version, the 100,000 base pairs. The second row is 10,000 base pairs. The red line, and then, the columns, the first branch length, the second branch length, and the third branch length. And the red lines correspond to the true values. And so what you see is that the estimates appear to be very nicely centered around the true values. And it actually looks, I don't know, for me, this is a really nice statistical problem, as coming at it as a statistician, because we have IID data. We might have whole genomes, so we actually have a lot of IID data, so the asymptotic should apply. I would actually expect these estimators to be asymptotically normal, and it looks like that could be true. And it looks like they're unbiased estimators as well. So that looked pretty good. And actually, it sort of gets even better, because if I look at these, so when I think of an estimation problem, I like to estimate the speciation times, right? But I'd also like to estimate uncertainty. I'd like to know, how confident am I in that speciation time? I'd like to form a confidence interval, perhaps. And so when I look at the form of the estimators, it's really cool because I just have a sum of things, a sum of random variables. So I can find the variance of that quite easily. I just sum up the variances and then worry about all the covariances. And in fact, since we're just viewing this as a sample from the multinomial distribution, we actually sort of know how these estimators should behave. As I said a minute ago, they should be normally distributed if my sample size is large, with this mean, this variance, and with this covariance. And that means then that I can just sort of, again, write an expression for the variance and plug in the observed values. Okay, and so here's, they're long, the expressions are long, but that's okay, right? No problem, still quite easy to evaluate. So the variance of that thing ends up looking like this, and we can just plug in the observed value for the variance. So sigma i's here are variances, so we can plug in the observed value for the variance. The sigma ij's are covariances, we can plug that in, and so we can get an estimate of the variance. So so we did that for the simulated data just to see how well this performed. And same setup, so more data at the top, the 100,000 base pairs, lower amounts of data at the bottom, so 10,000 base pairs. And then the variance of the first branch length. Oh, sorry, it looks like that's not true. Sorry, I'm telling you the wrong thing. This is the estimated values, and these are the variances. Sorry, I need to read the heading on my own slide. And I'm not sure which one this is. It's probably the larger sample size. In any case, you can see that the variance estimation works well too. So the true value is with the red line, and you can see that in this case, the standard deviation, they're distributed nicely around the true value so that the variance estimation works well also. Okay. Other fun statistical things you could think of doing, since these are expected to be asymptotically normal, we could think of making the standard confidence intervals that we would like to make. So we could take our estimate, add and subtract the right value from the normal distribution times the square root of the standard deviation, and that will give us the 95% confidence intervals. So we did that. We made 95% confidence intervals for the simulated data, and then looked at what proportion of them cover the true value. So these are plots of that. So these are the transformed branch lengths x1, x2, and x3. And you can sort of see that and the red line is the true value. So in this case, 94% of the intervals cover the true value, here 94%, here a little bit lower, 89%. But in general, we've looked over lots of simulations at this point. In general, we get pretty close to the expected 95% coverage for the confidence intervals. So it seems like things work, as we would expect in terms of estimating the speciation times. Okay, so that's great if you only ever have 4-tip trees, right? So I mean, I really like this problem because I think, it's a case again where in a lot of our statistics courses, we learn a lot about asymptotics. We spend a lot of time talking about asymptotics, and I feel like this is one problem where we actually kind of get into the realm where we can apply those asymptotics, because if I'm viewing each column in an alignment as an IID observation and I have a genome, then I really do have a large sample size. So the asymptotics apply, everything's fun. So I like that 4-tip setup a lot because of that. But if I ever want to do a bigger problem, I've got to do something else because I can't get the site pattern probabilities for more than 4 tips, or at least we haven't figured out how to automate that. Okay, so the question is, what do we do then when we have more than 4 taxa? And if you sort of draw out some of these trees, what you'll notice is that there are speciation times that appear in more than one set of 4. So for this tree, for example, I've highlighted one set of 4 that includes this particular speciation time. So pretend these are species trees for a minute. I got lazy and didn't draw the outline, but I mean these to be species trees. So this speciation time appears for this blue set of 4, and it also appears for this green set of 4, right? The only change is that I've not included this blue lineage and instead included this lineage here. Okay, and actually there's lots of them. You can now start to see them, and they all have that same time. So presumably the kind of thing I just did is informative about tau for each of those. Except it's weird because they're not like independently informative because they're sharing some of the tips, but they each have information. And so it seems like somehow you should be able to use that information together to estimate the species tree. So our first idea for this was, try to just get the estimates from every quartet that's relevant for a particular speciation time, and then try to compute a weighted average. And my idea there was that, these are unbiased. I think you could prove that they're asymptotically unbiased. So if you, just take a weighted average of them, that will also be unbiased. And then you get to pick how you take the weights. So just write down some math and try to figure out how to pick the weights to minimize the variance, right? That seems like you should come up with a good estimator. So, I had some grant money and hired a student, and that just didn't work at all, actually. So it turned out that the weighting part was really hard. So they were unbiased, that part is true. But the weighting part is really hard. We couldn't see very strong patterns in how to choose the weights to minimize the variance consistently. So, you might think it would depend on depth of the tree or something, but there were all kinds of other things that seemed to affect what the variance was. And, it may be that it could be done, but we couldn't see how to do it. So we decided to try something else. And that idea is to use the pseudo likelihood. And so I put a bunch of slides from different talks together. So sometimes I'm going to call it the pseudo likelihood, and sometimes I'm going to call it the composite likelihood. And I mean the same thing. So it's just not clear which word is the best, the best choice. So this will be pseudo likelihood / composite likelihood. And the idea here is that we're going to think of all the quartets in the tree, or we can think first of all the quartets that include a certain branch length tau. And we're going to write the likelihood for each of those quartets. So the I here indexes which of the quartets is being used to write the likelihood. The likelihood is just going to come from the multinomial distribution. So I can compute the site pattern probabilities for 4 tips, and that means I can write the likelihood function. Okay, so I'm using those site pattern probabilities from the first part, just not the formulas that I could get to estimate the branch lengths, but I'm still using the site pattern probabilities. So we can write the likelihood for particular quartets. And then what we're going to do is we're just going to multiply those likelihoods. Now, when I do that, I'm breaking a rule, right? We're allowed to multiply things when they're independent. Okay, in a probability framework, we can multiply if they're independent. These are definitely not independent because, like we said here, in the blue tree and the green tree, 3 of the 4 lineages are shared. So they're definitely not independent, but I'm going to multiply them anyway and pretend that they're independent. And that's why we call it either pseudo likelihood or composite likelihood. It's not a real likelihood. It's either a composite of a bunch of pieces, or it's a pseudo likelihood. It's a fake likelihood because we have this independence assumption. But still, the logic of it, I hope, makes sense, because if a particular value of tau is highly likely for all quartets, that should be a good tau. And if there's some tau that, isn't very likely for one of the quartets, that should bring the likelihood down. So this product of these 4-tip likelihoods does contain information about tau. And we'll then use the maximum likelihood estimator based on this composite likelihood as the value of tau that we pick for our estimator. And we can show that it's unbiased. I have that on this slide, but I'll try to show you a little better in a minute. So here's the idea. If we have a 5-leaf species tree, for example, we can break it. We can look at all possible 4 taxon subtrees. And then for each of these, we'll compute a likelihood for whatever branch lengths are in that tree. So this is a little bit of bookkeeping, because what you'll notice is not all times appear in every tree. So tau 1 is not included in this tree, for example, and tau 2 is not included in this tree. So, to implement this in the computer is not the most fun thing to do, but you can keep track of all of it. So we'll pull off all the quartets and then use what I'm now going to call composite likelihood to do the estimation. We actually made it a little more complicated than that to help with the numerics. So this is the actual formula. It's probably not important to see all of it, but this first part is just we took the log from before. So this is the log of the products of the likelihood. So that'll be the sum of the logs across all the quartets. And then the other thing that we changed was we added prior distributions on the age of the root and on the parameter in the multi-species coalescent. Because without the priors, the numerical optimization wanted to like go off and explore crazy spaces when we were trying to run the optimization routines. So the priors help stay on realistic values. And so that sort of helped for the numerical part of things. So in the end, what we call these estimators is our maximum a posteriori composite likelihood estimators. And to avoid saying that too many times, I'll just call them the MAP-CL estimators. And Dave Swafford has done something really awesome by implementing these in his software Popstar. So he calls them the CAGE estimators, so QAGE. It is computationally efficient and really slick to get these estimators in Popstar. So Dave's done an amazing job of working out all the numerical stuff going on here. So, so the advantage of these is that they're really computationally efficient, and I'll show you some timing data in a minute. But the disadvantage is that we treat the individual components as independent when they're not. And so the question is, you know, can we show anything about the mathematical properties of these estimators, given that we made this assumption of independence and they're really not independent. And it turns out that there's a really nice paper from the early 90s that can be used to show that these composite likelihood estimators are still statistically consistent and asymptotically normal. So I have all the math here, but you know, it's just the standard thing that you would expect, that when, as the sample size gets large, they converge to their true values, and you can find an expression for the variance. And so we actually have really good statistical properties for these estimators as well, which is great. Okay, so we wanted to, again, do some simulation to see how they work and compare them to other methods for this problem. And so, as I said at the beginning, since we can't compute the site pattern probabilities for more than 4 tips, there are no other likelihood-based approaches to this problem. So you can't get MLEs of the speciation times. But you can think of estimating the speciation times in a Bayesian algorithm. If you treat your state space as being composed of both the gene trees and the species trees, so that you search over that larger space, then you can run a Bayesian algorithm. And so that's what's done in StarBeast, for example. And then what we're going to talk about here is comparing to BPP, Zhiheng Yang's program. And so there are Bayesian versions of this. And so we were sort of trying to look, you know, both BPP and our method, the MAP-CL method, we use prior distributions. So we wanted to look at the effect on the effect of the prior, the accuracy of the estimation, and then how long it took to get the estimates. So those are the kinds of things that we wanted to compare. So we actually considered 9 different prior combinations. And so what's plotted here are all of those 9 different combinations. The 3 plots are different priors on the coalescent parameter theta, and that was from an inverse gamma distribution. It's not important what the values are here. The one in the middle is the centered at the true value of theta that generated the data, and this one makes thetas too small, puts more weight on small values of theta. This one puts more weight on large values of theta. And then the different colors in each plot represent different prior choices for the root age. And green, so the true value is blue, values that are too small are green, and values that are too large are red. And then the plotting symbol is the method. So the triangles are our method, MAP-CL, and the circles are the BPP. And the y-axis is the RMSE, the root mean squared error. So lower values are good. So the first thing, you can maybe look at this right away and say like, wow, oops, you can see it. Wow, you guys did a lot better than BPP. And it is true that our RMSE is below BPP's. But if you look at the y-axis, these numbers are tiny. So BPP also did a really great job. So you know, this is not really comparing the accuracy as much as just reassurance for us that our estimators are accurate. And so while we're, you know, a little bit better, both methods do a good job for the simulated data. And the effect of the prior, it looks like, you know, getting theta small is kind of bad for BPP. They do a little worse when the theta was underestimated, but otherwise it's pretty consistent. The priors didn't have a big effect on our performance. Oh, sorry, and the x-axis is the number of tips, it's the size of the tree. So the priors didn't have a big effect on how our method performed, a little more of an effect for BPP. And then in terms of timing, it's hard to make a timing comparison because BPP is an MCMC method. So you sort of run it until it converges. So it doesn't tell you when it should stop. So you have to make that decision. So it's kind of hard to figure out how to make a fair comparison. We tried, so we, you know, for different numbers of tips, we let it run, I think 50 times as long once we got into the larger problems. For the smaller problems, we were so quick that 50 times as long was still not very long. So we just sort of let it run for a really long time for the smaller problems. But what you can see here are the running times in this is like hours, minutes, seconds. So for the largest problem, we're getting estimates in 1 minute and 31 seconds. And to get sort of what we feel are good estimates from BPP was taking 27 hours. So there's a big difference in terms of time. And, you know, we could have stopped BPP earlier, possibly. And so, you know, that would maybe make it less accurate, but would reduce its time. So there's a trade-off there. But maybe a takeaway here is just that we can get estimates really quickly. We also looked at some of the trace plots to see if BPP had converged. And we do find lots of examples where there were some convergence problems. So even with fairly long runs, you know, 9 hours, 27 hours, there were some problems with convergence at some points. Okay, so I'll just wrap up with then an example for empirical data. So things usually work well on simulated data. If you simulate under the model and then estimate under the model, you would expect things to work really well. So at the end of the day, we want to make sure that when we have empirical data, which certainly deviates from the model some, that we still get kind of reasonable estimates. And so we used a data set for 5 species of gibbons. And there are some references here, and I tried to find a cute picture of a gibbon to show you, but probably others know more about these guys than I do. But this is a good test data set. There were over 11,000 loci, so over 2 million sites, so a nice long data set. And there were also multiple individuals sampled per species, so between 2 and 4 individuals in each of the species that you see here at the tips of the tree. And so the way that we handle that in terms of our quartet sampling is we take all quartets that have 1 member from each species. So we don't use sort of 2 members from the same species in the quartets, but we take all quartets where you could sample 1 individual from each species. And so that leads to 752 quartets. And so our likelihood then is a product of those 752 4 taxon likelihoods. That's our composite likelihood function plus the prior information. And so we first estimated the tree. So I didn't talk about it in this talk, but we also have a quartet-based method to estimate the tree called SVD quartets, and that took about 14 seconds to estimate the tree. BPP, in their paper, they ran it 10 times. 7 runs converged to the tree that I'm showing here, and 3 runs converged to a slightly different tree. But for the most part, we find the same for the same tree. Okay, so then on that fixed tree, we used both programs to estimate the branch lengths, and we made confidence intervals. For BPP, these are just HPD intervals, the posterior density intervals. And what we do for the confidence interval estimation is a bootstrap procedure. So just bootstrap the data set, and Dave Swafford's implemented the multi-locus bootstrap. So we can take into account the structuring in the loci, and then we get estimates of the variance that way. And so in this picture, so these are the 9 different priors that we tried. And number 5 is the priors that are centered on what we think are the true value. Well, but other people have estimated to be the true values, right? We don't know because it's empirical data, but probably the most appropriate priors are those in number 5. The red lines and dots are the estimates from our method, the MAP-CL method. So the dot is the point estimator, and then the interval goes from the endpoints here tell you where the interval goes. And these are for 3 different internal branches in the tree. And it's interesting. So one thing that you can see is that we have much wider confidence intervals than BPP. So BPPs are in black. And that's pretty much what you'd expect with the composite likelihood method, right? So we said that we gain in efficiency because we can compute this, but we do that by making an assumption that's not true, right? We multiply as if they're independent when they're not independent. So we've got to lose something by doing that. And what we tend to lose is sort of precision of the estimate. So we get wider estimates in this composite likelihood setting. And so you can sort of see that we're in general wider. But for at least these 2 branches, there's a lot of overlap between the estimates from BPP and from our method. An exception is this branch length, where there's not a lot of overlap, and actually both methods sort of consistently don't overlap with each other. So one method thinks it's here, another method thinks it's here, not much overlap, and that's pretty consistent. Whereas in other cases, BPP's intervals depend on the prior a little bit. So you can see that there's sort of, at least these 2 settings, there's a pretty strong prior effect on where the interval is for this particular branch length. Ours seem to be really stable across the priors, primarily because we're just using those to help sort of bound us and keep us from going crazy. Oops. But it's interesting that these disagree. In terms of computation time, it takes about 17 seconds for our method to estimate everything. And for BPP, we ran it for a long time because we had a lot of indication of lack of convergence if we didn't run it for a long time. So, some of the runs we probably could have stopped earlier. And so it's, again, not a very fair timing comparison because we decide when to stop it. But at least for some of the runs, it didn't look like it had converged even after a 2-week runtime, which is the max that we could do on our without special permission on our cluster. So, so there's a pretty big difference in terms of computation time, although it's quite likely that BPP would have been close to these estimates much more quickly. So again, it's sort of hard to make that comparison. Okay, so I said I'd tell you what everyone did. I think I told you Julia and I wrote down the site pattern probabilities, and we sort of did the first part where we were deriving the formulas. Dave has programmed all of this and improved it a ton by programming it and being careful. And I didn't tell you what Jing did. Jing's a PhD student here in biostatistics at Ohio State, and she was the one who was responsible for, well, she first experimented with this idea of weighting that I started with, and realized that wasn't going to work. And then she's the one who's done the proof for the theory. And she did an initial implementation. She proved the theory, and then she did an initial implementation of the method in R before we started collaborating with Dave. So Jing's done an awesome job on that project. And so I just want to acknowledge all 3 of them for great work on this. And then here are some of the references. We have a paper about this up on bioRxiv now, but it's the original submission, and it's changed significantly since then, and we're hoping to resubmit it soon. The election is interfering with that because Dave is obsessed with the election and not this paper. So as soon as the election is resolved, hopefully we'll be able to get that resubmitted. But if you're interested, I can send you a current version. So just let me know. And with that, I will stop, and I'll be happy to, I think I went really quick, but I'll be happy to answer any questions anyone might have.
Yeah, thanks for this great talk. Are there any questions for Laura?
So I have one, if it's okay. So if I understand it correctly, like you are given as input the topology of the species tree, but not the lengths, and the thing you can do is figure out the length. The answer to this might just be no, but is there any hope for doing anything if you're not given the topology? Like what I was thinking of is maybe like if you want to figure out like the distances, if you want to figure out an estimate for the distances between species and use that to reconstruct the tree, like.
Right. So yeah. So
is it just not? Yeah.
No, there's a whole So what I completely ignored in my entire talk is that there's this whole rather large effort in the research community and literature about how to estimate the species tree. And so, so there's sort of lots of approaches to that, how you would estimate the tree. One option is a distance approach, right? So you can compute pairwise distances and then use those in your favorite distance-based algorithm to estimate the tree. And that's sort of interesting. So you might think that these gene trees sitting inside the species tree would mess up your distances. But I don't, some others in the group might know better, but there's some evidence that something like neighbor joining works really well for this problem. And so lots of us, including me, I mean, I've spent the last, probably 10 years of my career basically working on the problem of how to estimate the species tree, right? And, have these elaborate methods and lots of coding. And then, there's some recent work that says just use neighbor joining, that works, that works really well. So, so I don't think I could explain exactly why that works well, but it does seem like there's some promise to using some of the distance-based methods. So I'm not sure what would happen in terms of getting the lengths from a distance-based method. I haven't looked at that at all, but maybe, yeah. I know it's a good question. And I skipped, so like I said, I've spent a lot of time working on estimating the species tree. So I think I was sort of eager to talk about something else. So I assumed the species tree was fixed, but it's actually a really interesting problem to estimate the species tree too. And this method that we have, SVD quartets, is, I think, a neat way to do that, mathematically, a neat way to do that. So that's an interesting problem. Yeah.
Cool. Thanks.
So Laura, I just had a question. In using that result of Arnold and Strauss to establish the asymptotic normality and I guess the consistency of that composite likelihood, I guess there's some sort of conditions that need to be checked. I imagine that's some sort of a generic result, is it? And to apply it to this situation, is it difficult to verify those conditions for your problem?
Yes, so it is, you're right, it is a generic result, and there's just a set of conditions that you need to check. And whether or not it was difficult is sort of probably, I don't know, probably you've all had this experience. After it was done, it didn't seem that difficult, but trying to figure out how to do it was tough. So we sort of sat there looking at this paper, Jing and I, sat there looking at this paper for a long time, and sort of you sort of have to define, just define a bunch of conditional things the right way to make it happen. And so we would try, let's define them this way. No, that doesn't work, it breaks this. Let's define them this way. Oh, that doesn't work, it breaks this. And then finally you figure out how to define them, and then it's all beautiful, right? So if we had started there, it would have seemed easy, but because we tried a whole bunch of things that didn't work first, so it is a long, pretty uninteresting proof to read. We had it in the body of our paper, which we're hoping will be in bioinformatics, and the reviewers said, take it out and put it in the appendix, which kind of, it feels hard because you worked hard on it, but it'll be in a shortened version of it will be in the appendix. But it's a big part of her thesis, actually, because I don't think it was trivial to show. On the other hand, I do think that result is pretty general. I mean, I think there are lots of situations that will fit into that, and they give some examples in the paper as well, some more traditional statistical problems. And so it is a very nice result. The other interesting thing about it is if you sort of look at this, you might think, well, here you have an expression for the variance. So why don't you just plug in stuff and use, instead of doing that variance thing, instead of doing the bootstrap to get the variance, why not just plug into this and get the variance? And we tried that. I thought that would work really well, and it doesn't. And the reason is because those p hats, if you have all these site pattern the probability of all four disagreeing, for example, in any problem you want to look at where things have recently and rapidly diverged, you don't get a lot of variation in your sequences. So your estimates of the site pattern probabilities are often very near zero. And then you put something like that in the denominator and square it, and the whole thing just sort of blows up. And so this formula seems really nice for estimating the asymptotic variance. I was super excited, and it turns out the bootstrap is far better because this is just numerically too unstable. So it's an interesting thing. We spent a long time thinking we were coding that wrong before we started thinking hard about why that shouldn't work, and it's just really hard to get it to work.
Okay, thanks. Great.
So Laura, I was wondering about the size of the trees you've mentioned. You said that your method is actually quite fast compared to some of the other methods. And I think what was on the slide was like trees up to maybe 20 taxa, if I remember this correctly. Does that mean because your method is so quick, you can really handle really large trees? Or are there any other bottlenecks? Like what about like trees with 100 taxa, for example?
It's a good question. I think that's sort of the next thing I want to try is see what happens for 100 taxa. I mean, I think the software will run and nothing will break and it will give you some answers. What I'm not sure of is what kind of data requirements you might have to get really accurate estimates. I mean, if you have a 100 tip tree, you have a lot of branches. And so I don't know how much data we might need to get really accurate estimates or how that's all going to work. But I think computationally, there's no real issue. One thing you will run into, I should say, at some point, it will be hard to pull off all the quartets, right? So right now, we just pull off all the quartets. At some point, you know, n choose 4 becomes a very large number, and even pulling them off and enumerating them is not going to be something that you want to ask your computer to do. So that, I guess, will probably be the bottleneck. I think before that computational bottleneck, there's a data issue to be examined. And so, yeah, so we haven't tried more than 20 tips yet. This is all pretty new, actually, like this summer new. So we haven't tried more than 20 tips so far. So I'll be interested to see how it works for very large problems.
Okay, thank you.
But the thing is, the other methods, the model-based methods, can't really get close to those problems at all.
Yeah.
You know, you're not running Bayesian inference algorithms on trees with hundreds of tips right now at all. So I do feel like, you know, we're getting to an area where you need something, and even if the confidence intervals are wide, it's more than you can get from other methods. I should say that, you know, these kinds of methods that I'm talking about, where we work with site pattern probabilities and quartets, they really work well when you have large data set size. So if you only have an alignment of 100 or 1,000 base pairs, this would be a really terrible method. You just don't want to do it because you can't estimate site pattern probabilities well with only 1,000 observations. You really need, you know, 10,000, 100,000. Whole genomes are great. So I think they're a nice complement to the Bayes methods, because if you have sort of shorter alignments or fewer genes and not so many taxa, you should run MCMC and get the Bayes methods. But if you have a lot of data, whole genomes, and possibly a lot of taxa, this is a really nice approach, because that's when the statistical guarantees will kick in and you'll have well-behaved estimators.